Good afternoon. Today we're going to talk about evolution of viruses, of course. We're going to talk about principles and concepts. And what we know about evolution today, of course, is founded on the work of Charles Darwin, who drew a phylogenetic tree in his notebook. This is one of his notebooks where it's so neat. He decided this is how organisms evolve, by natural selection and adaptation of the fittest for whatever the environment might be. Of course, he didn't know anything about genes at the time, but now we can interpret this in terms of genes. I love that he wrote, "I think," on this page, and then he drew his tree. A couple of years ago, I was at a virology meeting, and I saw a young lady with that tattoo on her shoulder. It's Darwin's tattoo. I think that's pretty cool. She let me take a picture of it, of course. And today we can make these phylogenetic trees using gene sequences. So Darwin made his tree by looking at animals and seeing how they were related. We don't have to do that anymore. We can sequence their genomes and make phylogenetic trees like the one on the right. We've seen a few of these already in this course. These measure the genetic distance between organisms based on the sequence. We use computer algorithms to compare the sequence, and we can identify those that are most closely related. And to construct these, you compare the differences in sequences of many organisms. Each division, each circle on this tree is called a node, and to the right of the node are viruses with common ancestors. We don't usually have the common ancestor, they are implied by this. And what happens is you have a common ancestor, and the two viruses, virus 1 and 2 in this case, have entered different environments, could just be a different host, and they've diversified, and their sequence is different enough that we place them on different branches. Now, the root of all these trees is on the left here, so that's the assumed ancestor of all of them, and we usually don't have such ancestors because it's far back in time and we don't have samples for that time as well. So this is a phylogenetic tree that we use nowadays using sequences. We'll see some of these today and also in the rest of this course. And of course, Darwin's principle of natural selection we can now understand in terms of the gene being the unit of heredity and mutation. So for example, we have host protein. Let's focus on a protein, not an entire host. It's adapted to an environment. Then the environment changes. If it's a virus, it's going to a new host or a new geographical location. And this protein is no longer well adapted to its cellular environment. It's maladaptive. But we know that all organisms make mutations during their reproduction. And so one of those that encodes a protein that's better selected is now selected for a better fit to the new environment. We then have what we call purifying selection, because all of the deleterious alleles that arose during mutation are gotten rid of. That's called purifying selection. And then, of course, positive selection is the passage of that well-adapted allele to many different offspring in the new environment. So these are terms that you will see often, purifying selection and positive selection, when we talk about evolution. Darwin would have loved viruses. He knew nothing of them, of course. They hadn't yet been discovered. But if he knew about them, he would recognize that they are the best exemplars of evolution by natural selection. And for RNA viruses, you can do an evolution experiment in less than an hour. They reproduce so quickly. He would have absolutely loved them. Today we're going to talk about evolution in terms of two general questions. We're going to talk about where viruses came from, but mostly we're going to talk about where they are going. What are the different mechanisms of evolution that lead to their diversification? Modern virology has played a big role in our overall understanding of evolution, just like it's played a role in understanding so many things about cells. And this is because viruses change along with their host populations. So hosts change, they from day to day change. Just you from today to tomorrow are going to be different. and there are many other changes at different levels, and virus populations are selected to be adapted to those changes. And of course, virus populations, not only do they evolve in response to changing hosts, which could be a new host as well, but they can select populations as well. And it's quite clear throughout history that populations have changed substantially because of virus infections. The introduction of explorers into the New World brought their viruses with them and wiped out many native populations who had no immunity to the viruses that were brought in. So if the native population can't adapt, it will be exterminated. Not just people, of course, but everything else out there. Public is always confronted with evolution. Even people who don't believe in evolution, it's happening right in front of their eyes. New viral diseases, AIDS, West Nile in the US, hepatitis C, Ebola, Zika, yearly influenza, common cold viruses, drug resistance, not just of HIV, but of all viruses. This is evolution. So next time you confront someone who doesn't believe in evolution, you can say, well, what do you think all these new viruses arising and all the drug resistance, not just viruses, but bacteria? What do you think is that? That is evolution. They won't believe you, but at least you can try. And it's right there in front of us. Viruses evolve faster than many people can comprehend. You know, evolution in most people's minds takes thousands of years. They don't understand that with a virus, it's happening in minutes. Today we're going to talk about 4 main drivers of evolution of viruses, and they're shown here. First, making a large number of progeny. You know, we make at best 1 offspring a year, and so we evolve incredibly slowly, but viruses can have generation times. Well, for bacteria, it can be less than an hour. They can make huge numbers of offspring. So large numbers of progeny, large numbers of mutants accompany that. We'll talk about the mechanisms of that today. Quasispecies effects. This is something we'll define. The fact that a virus is a population of mutants, shown on the right here. That's a quasispecies. And of course, all this numbering and mutation allows fertile grounds for selection. So those are the 4 main drivers that make new viruses on a daily basis. Viruses make huge numbers of progeny. These are just 2 examples to show you the numbers: hepatitis B virus and HIV. The half-life itself is amazing. HIV turns over every 6 hours in the infected individual, a little slower for Hep B. 90% of HIV turns over on a daily basis, and there are total production in blood on a daily basis is about 10 to the 9th infectious particles for HIV, 10 to the 11th for HBV. So when a host is making so much virus, contains so many virus particles, and of course then the host is responding with an immune response, that's grounds for selection. Immune response is one of the kinds of pressures that selects for viruses that have changed, but there are many others as well. Evolution is only possible when mutations occur. We're going to talk about that a lot today. Viruses mutate at every replication. That is because, as we've already mentioned, polymerases are error-prone, and that fact alone has actually been selected for, as you will see. Viral genomes are always mutating, and so it drives me crazy to see headlines like this: the Ebola virus is mutating. You can find this on a daily basis in the press for any virus. This virus is mutating, that virus is mutating. Now, what they mean is maybe there are amino acid changes, because not every mutation in a virus population will lead to an amino acid change. But people who write these articles do not understand it. Scientists have said that the virus has mutated. Really, that's novel. Wow, what a discovery. You should get the Nobel Prize for that. The virus is mutated. So I'm being a little silly, but the point is they're always mutating. Every second they are mutating. Whether or not that has a consequence is the real issue. And most people who haven't taken a course like this maybe don't understand the difference between a mutation and a phenotype. By the way, a mutation is a change in nucleic acid. Remember that from day 1. It is not a change in a protein. It is not a change in a phenotype. By definition, it's a change in the nucleic acid. We mentioned last time that RNA viruses are the kings and queens of mutation. The polymerases make lots of misincorporations, about 1 for every 1,000 to 10,000 bases polymerized. The average error frequency among RNA viruses is 1 in 10 to the 4 or 10 to the 5. And in a 10 kilobase virus genome, if the mutation frequency is 1 in 10,000, every time that genome replicates, you accumulate one mutation, and there are always thousands and thousands of new viruses produced. And so it's very easy to get collections of viruses which virtually every position mutated. And of course, there is no proofreading to fix any of these mutations. So once a mutation enters a genome, it's there unless it's lethal. Not every mutation is lethal, but many of them are, if you change the protein to something that doesn't work. So many of them will never see the light of the next cell, but others are remaining in the population. DNA viruses have much lower mutation frequencies because they have error correction, which we talked about last time. They have proofreading. They generate less diversity, and therefore they evolve slower. If you look at all the viruses out there causing human diseases, just human diseases, you will see that most of them are RNA viruses. There are lots of DNA viruses out there for sure, but the RNA viruses outnumber them, and I think the main reason, one of the main reasons, is because of their greater diversity. Here is a graph which displays on the x-axis genome size with mutation rate. And this is in substitutions per nucleotide per generation. So the upper left is the highest mutation frequency, and the lower right is the bottom of the graph is the lowest mutation frequency. The genome size goes from small on the left to very big on the right. So here on the right are the biggest genomes. Those are bacterial genomes, and they have a very low mutation frequency, as you would expect. And then the next entities are viruses with double-stranded DNA. Those are the blue circles. You can see a few of those there. They have lowish mutation frequency compared to the rest of the virus. And then we have single-stranded DNA viruses, which have a higher mutation frequency than double-stranded DNA viruses. The reason for that is not really clear. People are looking into that. We have then scattered among here, we're getting smaller and smaller now, viruses with single-stranded RNA. Those are the yellow circles, and retroviruses in purple. We have a double-stranded RNA virus here. You can see they're scattered. So this particular plus RNA virus has a very high mutation frequency. And then the highest mutation frequency of all is this point up here in the upper left called a viroid. We're not going to talk about these this year. You can blame the snow for that. Usually we have a lecture on viroids and other really unusual agents, but the snow interfered. Viroid is a piece of RNA, about 100 to 300 nucleotides in length. It's single-stranded. It doesn't code for any proteins. And these are typically pathogens of plants. They can move from plant to plant. They get in the cell. They're replicated by Pol II, and they don't make any proteins, yet they can cause serious plant diseases. And they also have the highest mutation rate. They use, which is weird because they use the host polymerase, but they still have a high mutation rate. Next, we have to talk about a quasi-species, which was something discovered 50 years ago this month. Actually, it was published in Cell, and this was a paper where they studied the RNA of a bacteriophage called Q beta. We did not have sequencing at this time. That was 1978. It was just being developed, but they hadn't applied it. They used other ways of looking at the variation. And I remember when this paper was published, I had no idea what it meant. It was called Nucleotide Sequence Heterogeneity of an RNA Phage Population. And what they wrote is a Q beta phage population is in a dynamic equilibrium with viral mutants arising at a high rate and being strongly selected against. The genome cannot be described as a defined structure, but rather as a weighted average of a large number of different individual sequences. 40 years ago this month, not 50. So I didn't know what this meant. Most virologists at the time didn't appreciate it. It took me many years to understand what they were talking about. But basically, what the outcome is that we found for this virus and subsequently for other viruses, all other viruses, even DNA viruses, populations exist as non-identical but related replicons. A replicon is simply a replicating nucleic acid. And we call this a quasi-species. This is not really a good name because of the species concept, of course, means organisms that can't reproduce. They can't make fertile offspring. And I don't know why that applies to viruses at all, even though some people call viruses species at a certain level, right? But then to say quasi-species confuses it even more. People are getting away from this term, but it's still in the literature. People are using terms like mutant, swarms, and some other things. But I will use quasi-species even though I don't like it. So here is a quasi-species on the left. Here are each line is a viral genome. Could be polio, could be any virus. And the different symbols show you different changes of one genome versus the other. The point here is that every genome is pretty much different. They have different kinds of base sequences. On the right is the sequence of poliovirus, which I did as a postdoc in 1981. And this was one of the first viral genomes to be sequenced. I think I've told you before, it took me 1 year of my life to do this, which now you could put it in an envelope and have it done in 10 minutes, but that's the way technology goes. So I did this sequence and I said, wow, I got the polio genome sequence. No, it's not. It's not one sequence. This is a consensus. What I sequenced was this mixed population, and what I have is the average of that population in my sequencing, because the technology at the time couldn't detect individual genomes. Now you can do deep sequencing and detect individual genomes in a virus population. So you can actually sequence the quasi-species. So whenever you think of a virus, remember, it's a mixed sequence and not a single one. And that has enormous implications for evolution, as you'll see. And why that is because when a virus infects a cell, no matter if it's a single-celled organism or a multicellular organism, it's always initiating with a population of particles. When influenza infects us, we're getting infected by a quasi-species, many different viral genomes. And whatever comes out of our cells are products of selection in those cells. It may not be the same as what went in. The viruses that come out of a new cell, the survivors, can then infect a new host, and that host will place selection on them. So you can imagine that every time this quasi-species goes into a cell in a host, something comes out that will then be different when it comes out of the cell of another host. Even two humans, the pressures are going to be slightly different. This is what I call the myth of consensus genome sequences. The myth is that sequence in the back. For a given population, there is an average, so that if you do average sequence methods, you will get this average sequence. But really, every genome is different. And in fact, this consensus may not even exist in the population. We are still working on understanding this. The problem with deep sequencing, which can be used to enumerate a quasi-species, is that it's error-prone in itself. Deep sequencing methods do not give you error-free sequencing. So it's hard to separate the error from the actual quasi-species. People are trying to develop methods to do this, and some people think they have. So we're still in the process of actually describing the quasi-species for individual viruses. But the main point is, it is clearly a mixture of genomes, and what goes into a cell can be very different from what comes out. So the quasi-species diversity is caused by the error-prone polymerases. The quasi-species are more diverse in an RNA virus population than in a DNA virus population. But there are also other contributors to diversity besides mutation. They're shown here. Recombination in co-infected cells. Remember, genomes can be copied by polymerases, and the polymerase can go from one to another to make recombinant genomes. So that's recombination, or reassortment. If you have viruses with segmented genomes, co-infection of a cell can result in reassortants, which have segments from both parents. So these contribute to the quasispecies. So now when we infect a cell with a quasispecies, complicated things happen. So here we say survival of the fittest. So in this genome, in this quasispecies, maybe one genome here, the star-star triangle genome, maybe that's the best to infect a bat that the virus happens to get into. And that will be selectively amplified, and as it multiplies in the bat, it will generate a quasispecies, so it will not be the only genome that is present. And so we have selected out a particular mutation or set of mutations, which will then be found in all the progeny genomes. So that's survival of the fittest. That goes back to Darwin, but now we understand at a molecular level what's happening. But the unselected mutations will also get a free ride. So maybe only star 1 is needed to replicate in the bat, but star 2 and triangle goes along, and maybe there are other genomes that happen to go along with it that are reasonably fit. So we call these unselected hitchhikers. They get a free ride. Survival of the survivors. So those viruses are not selected for, but they come along with the survival of the fittest. So after replication occurs in a host, and again, this can happen in a single infected host as the virus moves from cell to cell. You have a new population based on selection and the mutations that come along. A recent study last year showed that when mosquitoes take up virus, they inject into the host a very restricted population of quasispecies. In the host, this expands. When the next mosquito picks up virus from that host, it then restricts the population as well. So it can also contract and expand. This whole idea of mutation rate is a property that is selected. You may think, well, why don't polymerase evolve to be perfect? And in fact, we can make in the laboratory RNA and DNA polymerases that make a lot less errors. This has been done for both viruses and host cells. You can make mutations that reduce the frequency of incorporation errors. Here's one here. This is a famous one. Well, it's famous to me because it's poliovirus dependent RNA polymerase. It's glycine 64. It's a position in the active site of the polymerase. And that single amino acid reduces the error rate of that polymerase by many-fold. These lower rates are never selected in nature. We never see polymerases with lower mutation rate selected either in viruses in nature or in hosts. If you make them in the laboratory, it's relatively easy to select for such viruses and study them, they're often less pathogenic. So if you take this poliovirus mutant and put it into a mouse, it's not very good at causing disease, whereas the wild type, which makes the normal number of errors, is. So that tells you two things. First of all, high mutation rates are selected during evolution of both viruses and cells. Mutation is good. It's a kind of a weird concept for most people, right? Because we associate mutations in humans with bad phenotypes, but that's just the ones that we're seeing. There are many others in us that are good, and there are many in viruses that are good as well. So diversity is a selected property, for sure. Another important concept is that we've been talking so far about how mutation is so great for evolution, but obviously there's got to be an upper limit of mutation, right? Because if you mutate the genome too much, it's going to be non-infectious. That is what I mean when I say error threshold. It's the upper limit of mutation beyond which the virus is no longer infectious. If you go over the error threshold, the genome is no longer infectious. But if you go below a certain amount below the error threshold, you cannot compete because you're not making enough mutations. And I just showed you that in the previous slide where we looked at polymerase mutations that reduce the error frequency. So there's obviously a sweet spot. And RNA viruses evolve very close to that error threshold. I'm going to show you an experiment in a moment that proves that. DNA viruses exist way below it, and probably because they don't make a lot of mutations compared with RNA viruses. So again, the error threshold is the upper limit of mutation. Beyond it, the virus is not infectious. Too far below it, it's also not able to compete in a host. Now here's an experiment with a DNA virus that shows you the error threshold. You take a cell culture infected with a DNA virus and you add a base analog, 5-azacitidine. Here's 5-azacitidine. If you remember any of the discussion last time about antivirals, you will recognize that it's a modified base. It's a modified C, in fact. And what happens is this is incorporated by the DNA virus polymerase as a C. So it doesn't cause chain termination. It's incorporated, the chain continues. And then when that strand is copied, the C looks like a T to the polymerase because it's chemically modified as AZA-C, and that causes G to A mutations or transitions. So what this compound does is causes mutations to be introduced in the genome. Wherever there is a C, this will be incorporated. As the DNA replicates, the C looks like a T, and the polymerase will put in an A. So when you do this, when you grow up DNA viruses in the presence of this drug, the mutation rate increases several orders of magnitude. That means 100 to 1,000-fold high. You get far more mutants. When a similar experiment is done with an RNA virus, the best you can do is increase the mutation frequency 2 or 3-fold, compared with 1,000-fold with a DNA virus. So the RNA viruses are existing right near the error threshold, because if you mutagenize their genome in this way, you can't go more than 2 or 3 times more mutations. Whereas with the DNA virus, you can put 1,000 times more mutations in the genome, and the virus will still be able to replicate. They haven't exceeded their error threshold. Here's the experiment with an RNA virus, and this is using an antiviral called ribavirin, which has been used for many years. And last time I told you that it was the only thing to use for hepatitis C virus, ribavirin plus interferon for many years. We didn't know how it worked. And now we understand that ribavirin is a mutagen, very much like 5-aza-C is a mutagen for DNA viruses. This is a mutagen for RNA viruses. It's incorporated by the polymerase, and it causes mutations in the genome. So here's an experiment where we've taken poliovirus, and we are adding increased amounts of ribavirin to the culture, and we're taking a virus that grows and looking at its infectivity, and then sequencing the genome and figuring out how many mutations are present per genome. So y-axis is the infectivity, whereas unmutagenized virus is 100%. And then we add more and more ribavirin, and we get 2 and 3 and 7 and 15 mutations per genome. And you can see the infectivity drops dramatically with just 2 mutations per genome introduced by ribavirin. And then you add 7 mutations per genome, and you've really lost all the infectivity. So this is the experimental evidence that RNA viruses are living right at the error threshold, right below it, because you can't mutagenize them very much. Remember, with the DNA virus, you could put lots more mutations in the genome. So that's the error threshold and how DNA and RNA viruses differ as to where they are in relationship to it. Another important concept we call a bottleneck. And now what I want to do for the next few slides is to explain how quasispecies relate to genetic bottlenecks. So here's an example of a genetic bottleneck. We take an RNA virus and we plaque it out. And so here, you remember plaque assays, you can take a pipette and plunge it into the agar and pick the virus there, and then you can take the virus in that plaque and replate it again and get more plaques, and then you can pick another one. You keep doing that over and over again and keep going from plaque to plaque without expanding each plaque. In other words, without taking each plaque and infecting monolayers. You just do plaque assay to plaque assay. And what you find is that if you do this 20 or 30 times, your virus can barely grow anymore. They're less fit than the original population. And so what's going on? It's the same cells. It's you doing the plaque assay. All your reagents are the same. There's no variation. How is this consistent with Darwinian selection? We haven't changed the environment. All we're doing is going from plaque to plaque. So what is going on? Well, what's going on is that we are subjecting the virus to a genetic bottleneck. We restrict replication to the viruses found in a single plaque. And there aren't so many viruses in a single plaque compared with an entire virus population. A few thousand viruses derived from our initial stock. So let's say we're doing this with polio. We have a stock of virus which is 10 to the 9th PFU per mil. And we infect the monolayer with that, but we're restricting the passage to a few thousand progeny. And so if you look at the bottle metaphor here, each colored bead is a different virus, a different genotype within the quasispecies. Only a few can get through the bottleneck to go from cell to cell. And so we have a restricted population compared to the original total virus stock. So by plaquing from plaque to plaque, this is what we are doing. We are restricting the initial plaque to a very subset of the initial virus, and that's what we are then passing from generation to generation. So what is happening is explained by a genetic concept called Muller's ratchet. And this was formulated a long time ago by Dr. Muller to explain why sex arose in organisms. And what he found was that small asexual populations, organisms that do not reproduce sexually, just by division, they eventually will accumulate deleterious mutations. And he said this is why sex arose in nature, to try and get rid of these mutations. And so in our case, we've done this experiment with an RNA virus. They are, remember, very close to the error threshold, and we're restricting population to single plaques, which have very restricted diversity. We are accumulating mutations at each point, and eventually we accumulate so many that the virus exceeds its error threshold. There are more mutations per genome than it can handle to survive, and that's why fitness in that experiment goes down. And the reason it's called a ratchet is because each mutation works like a ratchet. You know, a ratchet is a wheel that can go forward, but it can't go back. So you introduce a mutation into the genome by the polymerase, the ratchet goes forward, cannot go back. Another one pushes it forward, and you keep pushing the ratchet forward until you have so many mutations accumulated that the virus is no longer infectious. Now you're saying, why is this different from a normal infection where you're always accumulating mutations? The difference is that your population is restricted. There's not enough diversity so that you can pull out from the quasispecies a virus that would work, that would be more fit. So it's kind of shown in this picture. So on the left, we have our quasispecies of virus. And if we infect cells with this virus and let them replicate, the whole population is replicating. And the viruses that are fit for that day in that particular cell emerge and predominate. But when we pick a plaque, we are isolating the progeny of one of these genomes, essentially. Let's say star-star triangle again. And we amplify that. Now you can see the quasispecies is very different, and it's restricted. And because the population is so small, as we go through these plaque-to-plaque passages, we never generate the diversity to rescue the infectivity. And that's why that kind of experiment causes fitness to go down. And so here are 5 different examples of fitness decline after passage through a bottleneck. The bottleneck is plaque assay, plaque-to-plaque passage. We have bacteriophage, VSV, foot and mouth, HIV, and other bacteriophage. You can see the number of passages, plaque-to-plaque passages, and how much fitness declines after each passage. Again, in each case, because we are restricting the diversity of the population. Now, you may say, does this have anything to do with the real world? And I would say, yeah, probably. There are some examples of it. But we not we, the viruses can overcome this. And so aerosol transmission, when you get infected by someone talking and spitting out small droplets, those small droplets don't have many virus particles in them. So that's like a bottleneck, right? The difference is that the virus is then replicating in you and generating the diversity again, whereas we restricted it to a single plaque. So that's in part why it works. Activation of a latent virus from a proviral genome from a few cells, that would be another example. Insect bites, they don't put a lot of virus in you either. So how do the infections escape the ratchet? Well, as I just told you, they multiply in the host, but also we generate the diversity that the viruses require. And in fact, you can do that in your experiment. Instead of picking a single plaque, you can pick 3 or 4 or more and do the passage that way, and you won't get any of this restricted diversity. And so that gives you the diversity you need to overcome the Muller's ratchet. If a mosquito bites me, the virus will replicate extensively in me and generate the quasi-species over again. There are other ways of repairing or fixing Muller's ratchet, if you will. Recombination is another one. If the ratchet makes 2 sick viruses, those 2 can recombine in a host to produce a healthy viral recombinant. So there are ways in nature of overcoming Muller's ratchet. So the message is very clear. Diversity is important for the survival of individual members. That's kind of a tough thing, maybe not so straightforward. Survival of an individual member depends on the diversity of the population. If you restrict the diversity, the whole population suffers and no one can get through. So that is error threshold and Muller's ratchet. Now, mutation and selection are 2 of the important parameters for evolution. Let's look at 2 examples of selection in the real world to make it more evident to you what's going on. So let's talk about viral selection imposed by the immune response. And here it's called genetic shift and drift. We've talked about this a bit before in terms of influenza virus. Genetic shift and drift is the selection of mutants resistant to antibodies or cytotoxic T cells. So if you have a healthy individual that's infected with a virus, you will make antibodies against the virus, you will make CTLs, and that will apply pressure on the virus population to select for variants that can evade the antibodies or the cytotoxic T cells. And so this can happen in 2 ways. The variant viruses can arise by single mutations caused by polymerase errors, we call that drift, or they can arise by recombination or reassortment to give bigger differences among viruses, we call that shift. And let's look at that with influenza viruses. Remember, the influenza viruses have hemagglutinin and neuraminidase in the particle. These are antigenic proteins that give rise to antibodies and CTLs. And we've talked about H1N1 and H2N2. The reason we do that is that there are 18 different hemagglutinins that we know of out there and 11 different neuraminidases. So they can occur in different combinations. And that's why we have H1N1, H2N2, H7N9. You can find all of them out there. Only H1, 2, and 3 infect people, though. Now remember that the hemagglutinin itself has antigenic sites at its very tip. You make antibodies against these epitopes. Those antibodies block infection by neutralizing virus. So an influenza virus infects you. Let's say you've been immunized the previous season with a vaccine, and you're infected with an H1 virus, which you were vaccinated against. As the virus replicates in you, it's generating diversity by mutation. And maybe one virus arises that has a mutation in one of these antigenic sites that your antibodies are directed against. That virus will not be neutralized very well. You may not get sick, but you'll still replicate virus and spread it, and then the next person will get a bigger dose of the resistant virus, and eventually that will spread through the population. So that's what happens sometimes on a year-to-year basis. The immune pressure, the antibody pressure on the virus selects for variants that randomly arise by polymerase error that are less neutralized by the antibody. They spread in the population, and then we have to change the vaccine. And that's why we're trying to make vaccines directed against stem epitopes which do not vary as much. And probably they don't vary as much because the structure of that stem is important. So that's antigenic drift, single amino acid changes that allow the virus to evade neutralization by antibody. The other part of this is antigenic shift, which is a major kind of change that occurs, and that's illustrated here. I mentioned before that every 30 or 40 or 50 years, influenza viruses undergo major changes so that nobody's immune whatsoever. The virus spreads throughout the world, infects many people. Those are called pandemics. We had a big one in 1918 where millions of people were killed. Very unusual virus. It came from a bird. As far as we can tell, all of the genes of the human virus came from a virus that circulated in birds. This virus stayed around, and then in 1957, suddenly a brand new virus we'd never seen before in humans arose, H2N2. What that was a reassortant of the H1N1 virus with a different bird virus, H2N2, and this one has the yellow RNA segment, so you can see 1, 2, 3 segments came from that duck virus, and that created the new one. And that includes the HA and the NA genes, so no human had ever seen these HA and NAs before, so no one was immune, therefore the virus can spread globally. And then in 1968, the H3N2 arose, and again, it was a reassortant of the H2N2, and now it got one internal protein gene and the neuraminidase, and the HA gene from yet a different bird virus. So one of the points here is that there's a huge reservoir of influenza viruses in birds and other animals as well, and those reassort with human viruses. Where they do so, we'll talk about more next time, but we think it's in chickens and pigs, and the increase in farming of chickens and pigs has allowed for a lot of this reassortment to occur. More recently, the most recent pandemic of influenza was 2009. It was caused by a novel H1N1 virus, not like the one that emerged in 1918 at all. But it turns out that this 1918 virus, when it emerged in humans in 1918, it also went into pigs, domestic pigs, and it has stayed in pigs from 1918 to the present. It does not undergo much antigenic variation in pigs. One of the reasons is they don't live very long. I think the average pig, you kill it in 6 to 9 months when it's big. So you never really have a chance for this virus to drift in these animals. So one of these 1918 H1N1s that had been circulating in pigs contributed some genes to this. That's called, so this is the virus on the right that arose in 2009. Look at all the different colored segments. It got let's see, the classic swine H1N1, that's coming from 1918. It's been growing in pigs ever since, so it got 3 genes from that. It got 2 genes from Eurasian pigs. Eurasian pigs, these are pigs that go back and forth between China and Europe, and they're different from North American pigs. They have different kinds of flu viruses, so some of those genes went in. One human H3N2 gene went in, a couple of avian genes went in, and there was an intermediate. There were 2 intermediate viruses. You can see how all these things reassorted to form the final one. So where this happened, we're not sure, but it probably happens on pig farms in different parts of the world. And on a pig farm, there are people who take care of the pigs. They get close to them. They'll inhale the virus. They'll get sick. They'll go home and spread it, and boom. A brand new virus with HA and NA, unlike any seen before, and it starts a pandemic. So this is what I mean by antigenic selection. We have drift, small changes, or we have shift, where we have brand new segments put in that no one's seen before, and then we have big outbreaks. We don't have any vaccine against this. We have to make a vaccine quickly. So obviously, if we had a universal flu vaccine that would prevent any flu, this would be great. We wouldn't have to worry about these events anymore. Okay, now the other concept I want to address is what we call host virus arms races. And this is something we've only figured out because we can sequence and manipulate genomes. The concept here is we have a virus and we have a host defensive protein. It could be an antibody, it can be something on a CTL, or it can be any of the intrinsic proteins that we've talked about. But the two fit together nicely, and here the host is winning because the host protein can get rid of virus infection. But of course, because viruses undergo variation, certainly a variant will come up at some point that evades the host protein, and we say the virus is winning. But of course, the host can evolve as well, and it can change its defensive protein in turn, so the host is winning. So you get the picture. Over a long time, this goes back and forth, back and forth. That's why we call it a host virus arms race. Some people call it the Red Queen hypothesis because, the queen here, and Alice said around here, it takes all the running you can do to just stay in the same place. I think you can understand why it's called the Red Queen hypothesis. We can now tell you exactly what's going on in these arms races. Let me tell you how to do that. First, we can sequence tons of viruses and their genes and tons of host species. And so what we do is here we're looking at primates, different primates, to look for a host virus arms race in that species. And we look at a particular protein. We can look at many proteins, but what you do is you look at the sequence and you look for signs of what we call positive selection, which I mentioned before. If you compare different proteins, orthologs means the same protein in different animals. There are a lot of changes that in the sequence that don't make an amino acid change. We call those synonymous changes. And then there are changes, they're in red here, which are non-synonymous because they change the amino acid. And people have written computer programs which simply go through all these sequences and look at the ratio of synonymous to non-synonymous changes to get an idea of which protein changes are being positively selected in evolution. And you can then look at that and see, wow, there's some residues that are putatively positively selected, and you can map them to particular lineages, they're shown in red. And then you can go right down to the amino acid sequence and say, at this position in this protein, which we think is a host defensive protein, there is an amino acid that has been selected for at some point in time. That's the signature of virus exerting positive selection at a particular residue. Then what you can do, establish a cell culture system and test this idea. You can establish cells from your different primates. So right now we've got cell lines from almost every primate that's out there. You can take them with and without the selected changes that you've identified. Then you can infect cells and ask, does the virus replicate or not? And whether the virus wins or the host wins, right? So virus winning simply means it will replicate well in the cells. If the host wins, it means it's refractory. And then finally, you can take the changes which seem to go with virus or host winning, and you can introduce them into the host protein, and you can introduce them into the virus protein. So in this case, we have a virus which is winning in this particular host. We can take the protein and change it at that position that we've identified to be positively selected, and now we see the host is winning. So we can actually prove in cells this positive selection has led to a virus-host conflict. And this has been done in many laboratories now for all sorts of virus-host combinations. And the bottom line is really neat. Virus-host conflicts have driven the evolution of the immune system. It makes perfect sense, right? We get infected, we have an immune response, there's a conflict with the virus, that's going to evolve over time. So here are two proteins where I want to illustrate that. On the left is an intrinsic defensive protein called TRIM5 alpha, which is involved in defense against certain retroviruses. It has two domains. It has an RBCC and a B30.2 domain. The important point here is that there are positively selected amino acids. That's the inverted triangle. Very clear in this domain, and they were eventually mapped to an interaction with the HIV capsid. What this protein does, it binds the capsid of HIV and it causes it to fall apart. So there has been a continuous virus-host arms race between this protein and the HIV capsid. And all those triangles are individual amino acid changes that reflect that, which have been tested using the approach I've just told you. On the right is PKR. I don't know if you remember PKR, but this is a host protein that is activated by double-stranded RNA, which phosphorylates the IF2 alpha and leads to translational shutdown. So it's a host defense protein. And this one has, again, by sequencing the protein from many different animals, it has positively selected amino acids throughout its sequence in both RNA binding domain and in the kinase domain. And by testing individual viruses against this protein, we see that the RNA binding domain has been in conflict with herpes US11, a specific herpes protein, a specific flu protein, specific pox virus protein, and the other positively selected amino acids in the kinase domain have been selected by a variety of other viral proteins as well. So you can see the evolution of the host immune system and how the virus has compensated for that as well. So here we have an interesting situation where PKR has different parts that are reacting and interacting in a conflict with different viral proteins. Here's one last example of this, which is pretty neat. This is the transferrin receptor. It's a rodent transferrin receptor. Of course, transferrin receptor binds transferrin, which is a protein that carries iron into the cell. It's essential for us. It's on every one of our cells. It also happens to be the receptor for two different mouse viruses, an arenavirus and mouse mammary tumor virus. And one laboratory has identified the interaction sites of the virus with this protein. And for arenavirus, it's the gray sites. And for MMTV, it's the blue sites. So these are areas where there have been positively selected amino acids in the receptor for these viruses. You can see they're distinct, and they have changed over time as the virus changes. Remember, the virus binds this receptor. Receptor can change, or the virus can change to go back and forth in this arms race. So the point here is that the interacting protein doesn't have to be an immune protein. It can be any protein in the cell that's required for cell replication. In this case, it's a receptor. So you can imagine very easily cells evolve also, and if there is a receptor that arises that the virus can't bind to, that will predominate in the rodent population until it selects for a virus that can overcome that. And that's what these dots mean. These are the sites on the receptor that change, and the sites on the virus that change have also been identified as well. Importantly, these interaction sites for the two viruses are different from where transferrin binds. Transferrin binds down here. Of course, it makes perfect sense. You couldn't change the transferrin binding site because then you wouldn't be able to change transferrin. But the viruses don't bind there. They bind elsewhere. So we have a lot of genome diversity, as I've shown you in just a few examples. Yet, here's a conundrum. There are only 3 serotypes of poliovirus. There's only 1 measles serotype. Yet we see continuous influenza variation. We see 150 serotypes more of rhinoviruses. Why are these viruses different? I don't know the answer. It's a really interesting question. So clearly, some structural proteins of viruses are able to sustain more mutations than others. And polio and measles are very restricted. Flu and rhino are obviously not. We don't know why this is. It may have something to do with architecture, but I don't understand it because measles and flu are both enveloped, yet the measles glycoprotein cannot change at all to change serotype, yet the flu glycoproteins can. So this is a mystery that requires solving. The next topic I want to get into in terms of evolution deals with virulence, the capacity of viruses to cause disease. And the question here is virulence positively or negatively selected? What do I mean by that? Here's the hypothesis. The idea is that viruses shouldn't be too virulent because if they kill the host, especially if they kill it too quickly, there's not going to be opportunity for transmission to a new host. If that's correct, then all viruses should be very infectious and avirulent. That would be the trajectory of evolution. But we don't actually see that. We see that there are many human viruses out there that have been with us for a while, and they're virulent. So what is going on? Let me first give you an example of an experiment which supports the idea that there is an evolution of virulence to lesser virulence. And you have to keep in mind during this discussion that there are often virus jumps from species to species. So all the viruses we have came from someone else at some point, another animal or our ancestors, and they jumped into us. And so what we're talking about is the virulence when the virus first enters a species compared to many, many years later. Does it change to spare the host? And this is an experiment done in Australia. In Australia in 1859, they brought a rabbit in from Europe because they wanted to hunt rabbits, and they didn't have any in Australia. So they brought them in, and there were no predators of these rabbits, so they replicated like crazy, and there were tons of them. And so then they said, we have too many rabbits, we have to get rid of them, so they introduced a virus to kill them. And this is myxoma virus. It was released in the 50s to get rid of the rabbits. The natural host is the cottontail rabbit. It is spread by mosquitoes. These cottontails get superficial warts, but European rabbits, which were introduced into Australia, they're not the natural host of this virus, so they're wiped out by infection. So this is a great example of a virus jumping to a new species. In this case, we made it jump because we introduced it to wipe out the European rabbits. The virus has been existing with cottontails for many years, so it has apparently evolved to be less virulent, or so we think. So the first year they released this virus, it killed 99.8% of the rabbits. But then the second year, the mortality dropped to 30%. So those 0.2% of the rabbits had some mutations in them that made them more resistant, and the virus also changed to be less virulent. Then after that second year, the rate of killing was lower than the reproductive rate of the rabbits, and they could never eradicate it. So this is a failure because both the host and the virus evolved to a point where the virus was not killing all the hosts. I think it makes sense that that would happen. Otherwise, there's no host left for the virus. Now, of course, the virus is not thinking this at all. The virus is not thinking, I can't kill this host or I wouldn't have any left. Of course not. It's just evolving in a way that's maximally beneficial for the moment. So how that actually happens is a good question. Some people think that the virus became less virulent, or you selected for virus of less virulence because those would be more likely to be picked up by mosquitoes. Because if you kill the rabbit quickly, the mosquito is not going to pick up the virus. So if the rabbit lives longer, better chance of a mosquito picking it up, and that's the selective force for whatever virus mutation is causing less virulence in the rabbits. So the end story here is that both rabbits and viruses reproduce to quite high levels. So this can happen very quickly. As I said, the virus and the rabbits evolved. And this is what we would predict. Initially, you put a virus into a new host, it kills a lot of them, but not all of them. Then it will evolve to become less virulent, so both the virus and the host can exist. Now, just as a side note here, Australia seems to not learn lessons because a few years ago they wanted to use herpes virus to wipe out carp. So a number of years ago, they imported carp to grow in fisheries. They had a flood and the carp got out into the rivers and they overgrew, and the rivers are full of carp and they don't like this. So they want to kill them with a herpes virus. But now they're saying that it may not work. And I would bet that if they tried it, it wouldn't work anyway, despite their prediction. What about people, though? What can we say about the evolution of virulence in people? We can't say anything. We don't have the beginning and the end like we did with the Australian experiment. We know that there are lots of virulent viruses, Lassa, Ebola, HIV. But these represent very recent jumps of the virus from animals into people. Whenever there's a Lassa or Ebola virus outbreak, it's a brand new transfer of virus from an animal in the forest to people. And it's very virulent, but it's just the beginning of that evolutionary process. HIV only jumped into humans in 1920, so we haven't had enough time to see the evolution of virulence. Now let's look at measles and polio. These have been in humans for tens of thousands of years. They probably were in our ancestors. They seem to be less virulent. So that would support this idea of evolution. But the problem is we don't have the first isolate of polio. Maybe it was avirulent or less virulent from day one. So we can't really conclude anything about human evolution of virulence. Nevertheless, the press is obsessed with it. Here are some headlines. Ebola is mutating and will go airborne. Is Ebola getting better at replicating? It goes from person to person. And then Peter Hotez said Zika got more virulent as it spread. None of these make any sense to me. Why would the virus get more virulent? If anything, it's going to be less virulent. And anyway, you can't tell anything from these studies, because in the end, you can't put the virus back into people to test your theory, so you're never going to know the answer. We have no data on the effect of evolution on virulence of human viruses. So next time you see a headline that says the virus is evolving to be more virulent, do not believe it. Let's end with a consideration of where viruses came from. For many years, we had no clue because we didn't have fossils like we have with other animals where we can date them and build phylogenetic trees. The oldest stocks we had were 1918 influenza virus, which we reconstructed, 30,000-year-old giant virus, which we recovered from Siberian permafrost, but that was really the oldest virus we had. And that doesn't really help you with evolution, where we're talking about 4 billion years of development of life on Earth. But when we got the ability to sequence genomes, then we could start saying, how fast are these genomes replicating, and can we backdate their existence by the mutation rate? And that's called molecular clocks, relating the time scale of viral evolution with that of the host. And so from that, we said herpes viruses must have infected the dinosaurs, because they were probably around 180 to 220 million years ago. But nowadays, we can do even better. In every cell, there are integrated DNA copies of different viruses, not just retroviruses. These are called endogenous viral elements, or EVEs. They're just pieces of influenza, measles virus, reoviruses that have accidentally been integrated into host genomes. And every animal on the planet has these. They don't seem to be of any consequence, but they allow us to date when these viruses circulated, because you can construct phylogenetic trees. Look at this one for rabbits. Here we have 3 different species of rabbits. They have a common ancestral host. We can tell that these 2, for example, have an integrated Eve, and we can see it's in the same place on the chromosome, so they probably got it from an ancestor. The 3rd rabbit does not have it. So in this way, we can date the insertion of the Eve, somewhere between the break between the species and the fixation in these 2 species. This is called phylogenomics, and we can say, how long ago were viruses around? So here's an example of doing this with a couple of small single-stranded DNA viruses in a variety of species. We could look in their genomes. We've arranged them in a phylogenetic tree so we know how they're related. We can tell where their ancestors branched off. So we can date the insertion. And we can say that in some species, 60 million years ago, these viruses were circulating. So that is really neat. We were never able to do that before. This has now been taken back 450 million years ago. They have found evidence of retroviruses in the genomes of some very old organisms. These estimates give rise to the idea that these retroviruses probably arose 450 million years ago in the Ordovician period. And so that is right here, the age of invertebrates. They may have arisen in the oceans, in organisms like this one, a nautiloid cephalopod, which is around at 488 million years ago. So that is the oldest dating of viral presence based on these methods that I know of. But I think probably viruses were around maybe in a different form before this, maybe billions of years ago. And here's one hypothesis for the origins of viruses. We start very long time ago in the RNA world where there were no cells. So this is called a pre-cellular stage of life on Earth. Many, many years ago, before cellular life, we have the evolution of self-replicating nucleic acids. RNA mainly initially, and these are called replicon. There are no viruses in the sense that we detect them today, but these are probably precursors of both viruses and cells. Eventually, and this is a big gap here, there is an emergence of primitive cells. So now we have a cell membrane, initially without a nucleus. These replicons start to replicate in these primitive cells. They give rise to proteins, of course. They give rise to reverse transcriptase, which now makes DNA, and you now have DNA cells. And at some point, these selfish replicons, we call them selfish replicators, capture capsid proteins, probably from the cell. So if we look at the structures of a variety of viral capsid proteins, we can see related proteins in cells. So the idea is these replicons grab the capsid protein gene and were able to escape and infect other cells. And that, of course, leads to the evolution of modern cells where we have a nucleus, which some people think was donated by a DNA virus. And viruses continue to evolve, of course. They didn't just evolve back a long time ago. They continue to evolve today. And we think that in some cases, the viruses start very small with minimal genomes and they acquired extra DNA by horizontal gene transfer. Or sometimes they can leave cells in a big way. So the giant viruses may be large viruses that escaped cells with lots of genes and then lost the ones they didn't need because they became parasites. So I think if you look at it this way, then viruses could have started to arise 4 billion years ago when the Earth had cooled and we think the first organic compounds were forming. Now all of our viruses, the human viruses, these probably evolved long before Homo sapiens was around on Earth. And we probably got them during our time as Homo sapiens or from our ancestors, which passed them on to us. And we got them from animals. So these, either Homo sapiens or its ancestors were infected with an animal virus, and many of these have stayed with us. We've lost a lot of them as well. I'm sure many dropped off along the way that we don't know about and will never know. But the human viruses we have now, many of them likely came from a long time ago, but others came sooner. Let's look at two examples. Here's smallpox. Sequence analysis of various isolates suggest recent introduction into humans. A close ancestor of smallpox is a virus that infects gerbils. And so maybe Homo sapiens acquired infection from gerbils at some point that remained in the human population and became the smallpox that we know. Now you have to remember that when you have small populations of Homo sapiens and their ancestors, they're not likely to pass viruses among them. They're typically very small communities that live apart from each other. So it's only when Homo sapiens begin to congregate in larger numbers are we really getting the kind of numbers we need to pass virus infections. So the smallpox, this could be 15,000 years ago that this happened. Measles virus jumped into humans about 5,000 years ago. It's very related to a virus of cows called rinderpest virus. And the idea is we first began to domesticate cattle when we were, the ability to domesticate animals made bigger cities possible because the food supply was assured. So we now had cows around us, and we got viruses from the cows. We domesticate animals, we get those viruses. So we think that measles came from cows to humans in the Middle East about 5,000 years ago when people began to congregate. And then colonization and migration spread it. It spread to Europe, and then Europeans went to the New World. They brought measles with them, and the Native Americans had never seen measles because they didn't have cows and no one had ever come before. So they got infected and wiped out because they had no immunity. Great example of introducing virus into a new population. So that's part of the evidence that measles was once very virulent and it has evolved to be less so. So what about new viruses? How do they arise? I haven't seen any new viruses in our lifetime because we just keep discovering things we haven't seen before. And so it's very hard to track if any of them are new or not. But the assumption is that a new virus will only arise from what's here. It's not going to just pop into being. So let's think about how many new viruses there could be. What's the number of all possible mutations of a viral genome? Because to get a flu to mutate into something else, you need mutations. So some studies have shown that half of the nucleotides in an RNA virus can accommodate mutations. So let's stick with that number, half. So a 10 kb viral genome, there are 4 to the 5,000 different possible sequences that can arise by mutation. 4 to the 5,000 and more by deletions and recombination. There are only 4 to the 135 atoms in the visible universe. 4 to the 135, yet there are 4 to the 5,000 possible different permutations of a viral genome, again, assuming that half of the nucleotides can mutate. So you can make a lot of different possibilities. But we don't see flu mutating into herpes virus. So there are obviously constraints that reduce this number substantially. Herpes is always herpes, flu is always flu. So then the question is, how do we maintain that stability? Or how do viruses stay flu? How does a flu virus stay flu, polio remains polio, and so forth? Well, there are many constraints on viral genomes. The genome itself, DNA can't turn into RNA. You can have lots of mutations, but DNA will always be DNA, and RNA will always be RNA. Replication is important, obviously, and genomes have to interact with viral proteins and cell proteins, so they can't change so much to lose those interactions. They won't be able to replicate. The nature of the capsid restricts the length of the genome. You know, a physical icosahedral capsid has a fixed internal size, so the genome can't get very big. And then, of course, the host. If a virus evolves to be too lethal, it'll wipe out the host and never become established. And of course, if the virus doesn't replicate well in a host, it won't be established either. So these are some of the constraints that we see on virus evolution that make all the viruses that are around today the main ones that are going to stay with us. Now, next time we're going to talk about emergence. And there, the key is that we do see new human infections, but they're all viruses that have been in animals already. And let me leave you with this slide, which I really love. You know, it took 8 million years for Homo sapiens to evolve. Our DNA is 98% similar to that of chimps. So it took 8 million years to have a 2% divergence in our DNA sequence. And poliovirus changes the same amount, 2% in its genome sequence, as from the time you ingest it to the time you excrete it. About 5 days, it changes 2% of its sequence. So think about what a virus could do with 8 million years.